DNA is composed of two strands of nucleotides wound around one another in a double helix. The outer longitudinal structure consists of alternating sugars and phosphate groups. Paired nitrogenous bases are in the interior of the molecule. If we unwind the molecule, its structure resembles a ladder with phosphates and sugars forming the sides. The rungs are the nitrogenous bases of nucleotides on opposite sides of the ladder. There are 4 different bases, and only certain pairs are possible in forming the rungs. Adenine is always opposite thymine, and guanine is always opposite cytosine. These complementary pairs of nitrogenous bases lie next to one another because their sugar-phosphate chains run in opposite directions. This anti-parallel orientation of the two nucleotide strands brings the bases into position for hydrogen bond formation. The sides of the ladder are twisted in a spiral fashion and make a complete turn every 10 base pairs.
DNA molecules are extremely long, thin, and delicate. They are packaged so that they can fit in the nucleus and be protected from damage. DNA is attracted to clusters of histone proteins. A portion of the DNA wraps around a cluster twice. This occurs at regular intervals along the molecule. This shortens the DNA so it can fit within the nucleus. The chromosomes are still fairly long, and their movement during cell division is facilitated by condensation. Following replication, they loop and fold back on themselves, forming tight packages of DNA and histone. This shortens and thickens the chromosomes, so they are visible under a microscope at the beginning of prophase. The rule of base pairing helps explain how DNA is replicated prior to cell division. Enzymes unzip the DNA by breaking the hydrogen bonds between the base pairs. The unpaired bases are now free to bind with other nucleotides with the appropriate complementary bases. The enzyme primase begins the process by synthesizing short primers of RNA nucleotides complementary to the unpaired DNA. DNA polymerase now attaches DNA nucleotides to one end of the growing complementary strand of nucleotides. Replication proceeds continuously along one strand, called the leading strand, which is shown here on the left. The process occurs in separate short segments called Okazaki fragments, next to the other, or lagging strand on the right. This difference is due to the fact that DNA polymerase can only add new nucleotides to the 3' end of a nucleotide strand. A primer begins any new strand, including each Okazaki fragment. An enzyme replaces the RNA primer with DNA nucleotides. Then an enzyme called DNA ligase binds the fragments to one another. There are now 2 DNA molecules. Each consists of an original nucleotide strand next to a new complementary strand. The 2 molecules are identical to each other.